this session is we're going to start with some case studies. We've got kind of two examples from the SolACE project, one in wheat and then also potato trials. And then we also have a bit of a critique of the approach that we used in SolACE, and then we'll kind of pause for questions. And then we also have some perspective on developing biostimulants commercially, and then examples of using arbuscular mycorrhizal fungi from an INTERFUTURE project, although we have had some technical difficulties. So I am going to need to make sure Miriam's all set to join us. We'll have a Q&A about those different topics, and we'll have a bit of a comfort break. And then we're going to jump into an interactive session where we hope we can continue discussing things. And then we've got a bit of an activity we're going to do together before the event comes to an end. So just to give you a bit of background, the SolACE project is specifically an EU project called Solutions for Improving Agroecosystem and Crop Efficiency for Water and Nutrient Use. Involves designing solutions and then using them to manage agroecosystems, and specifically focusing on crop efficiency in terms of breeding, and looking at its performance under stress. And just to give you the context of where this project has been taking place, we have partners across countries in Europe. There are research partners, industry partners, extension partners. We've been looking at organic and conventional growing, and mainly 3 different crops: bread wheat, durum wheat, and potato. And today specifically, I've pulled out some of the relevant objectives that we're kind of focusing on. So we're specifically looking at biostimulants, and we included microbial inoculants, where some of the innovations that this project we're looking at, evaluating the both in-field conditions and also on farm. So that's something we're specifically thinking about today, including looking at local solutions in these different countries. And then what are the ways that there are barriers to using these innovations. And then ultimately, we're hoping with this interactive activity, we're going to be engaging in discussion about what strategies work, what other questions do we have, and where do we go from here? So that is the introduction to the day and a bit of background on the project. And now I'm going to pass screen sharing on to Jean-Pierre Cohen from Arvalis, who's going to talk about examples from the SolACE project in wheat.
Okay, good afternoon, everyone. Do you see my screen correctly? Yes, perfect. So thank you, Amelia, for the introduction. So my name is Jean-Pierre Cohen from Arvalis in France. Arvalis is a French applied research institute funded by farmers in France. And And our objective is to design innovative tools and information for farmers. We play a lot of roles, and we have various involvement in project SolACE. But today I will show you what we did on microbial inoculant and the effect of microbial inoculant on wheat. My presentation will focus on result obtained in France, but I must mention at the very beginning of my presentation that we also work with our colleague from Agroscope in Switzerland to test some microbial inoculant in their countries. But I will explain why I will focus today only on French results. So this presentation was prepared with my colleague Gregory Verissel, which is the main architect of this result. So to give you a very brief introduction of what we could expect to use microbial inoculant in wheat, I like to show this triangle of nutrition improvement and mineral nutrition improvement with 3 summits. If you want to improve nitrogen or phosphorus uptake and utilization, you can play on 3 summits of innovation. You can work on recycling by using manure or urban waste, etc., etc. You can act on biological supply, I mean nitrogen fixation, either in symbiosis or in free microbia in soil. And you can also work on the main fluxes of nutrient by improving uptake and utilization of this element by studying soil properties and also to design some innovative fertilizer. And in this perspective, microbial inoculant could have some advantages to use in 2 summits of this triangle, the uptake and utilization summit, and also the biological supply summit. If we go deeper in the postulates and expectation concerning biological and microbial inoculant, I have to rapidly which type of bio-inoculant we use in the project. First of all, we studied some plant growth promoting either bacteria or rhizobacteria. This is a figure on the right of the slide. This type of microorganism could have a lot of interest to promote plant growth, of course, but also we could have some side impact on pathogens as a biocontrol effect. I won't go into all these details, but you can find a lot of information in the literature describing all the metabolic pathways we could trigger with microbial bio-inoculant in soil, but also in the plant. And the second main type of inoculant we studied in the project are arbuscular mycorrhizal fungi. This type of microorganism also have a lot of function and putative advantages for plant, but the main expectation is to extend the root surface to uptake more nutrient or water. That's the main feature of mycorrhizae. So in the project, we tested a mix of both type of microorganism. So we conducted some trials in France by using the following protocol. We tested 4 type of inoculant. The first one is very simple because we didn't apply any product. And then we studied 3 associations of microorganisms. The first one, I won't try to pronounce the Latin name of the organism because it's quite difficult. But to resume, the first one is a mixture of mycorrhizae and plant growth promoting rhizobacteria. The second one is also an association between mycorrhizae and bacteria, but also with another fungi. And the last one is another association of mycorrhizae and plant growth promoting rhizobacteria. So we tested all these microbial inoculant against several wheat cultivar, namely Rubisco, Cellule, and Cougar, which are used cultivar in the project, but also in the field in France. We would like to do it because it's very important to see if we could show some interaction between the genotype and the microbial inoculant, because in the literature, some interaction between genetics and microorganisms are expected. And the last factor we tested across all cultivars and microbial inoculant is the nitrogen rate. We tested 2 nitrogen rate, an optimum nitrogen rate, which is called X, and X minus 50 kilogram nitrogen per hectare, which is a low fertilizer treatment. The field trial network in France is composed of 4 trials, 2 harvested in 2019 and 2 harvested in 2020. As you can see on the map, we have 2 main experimental sites, one just below Paris, and one in the center of France, with some quite with soil with some similarities between sites, silty clay or silty clay loam, and various conditions concerning weather and agricultural practices. You can see my remark on this slide. 2 additional trials have been carried out by Agroscope in Switzerland in 2019 and 2020. The analysis is still ongoing with our colleague from Agroscope, and that's why I chose not to show this result, because some work are still needed to pull all the results in the same synthesis. Concerning the application of our microbial bio-inoculant, we did it at sowing on the seed bed. In 2019, we tested various formulations, and in 2020, all the microbial inoculant was applied with a mini-grant formulation from the DCM company. And I must underline that all the microorganisms were provided by partners from SolACE, which is very interesting because we have in the same project the producer of the microorganism, also the company which did the formulation, and also the partners which did the experiment. So we have all the information needed to correctly interpret the result. So first result, I will spend 1 or 2 minutes just to clearly explain how you could read these figures. So the comparison principle is to compare yield on the right side and grain protein content of wheat on the left side between the result obtained with the control, I mean with no bio-inoculant. This is the x-axis, compared to result obtained with microbial inoculant, which is the y-axis. So the black line is a bisectrix. So if the point is under the black line, it means that when you apply the bio-inoculant, you obtain less yield or grain protein content compared to the control. And if the point is above the black line, it means that the bio-inoculant have a positive effect on yield or grain protein concentration. And as you can see, we make some basic statistics to see if we could show some significant effect. And the first and the last information to keep in mind is that we present the result sorted by the treatment. In this slide, the results are sorted by cultivars. It means that the red, green, and blue colors are corresponding to different cultivars. The red one is Cellule, cougar. The black one is cellule and the green one is rubisco. So as the conclusion for this slide, we showed that we can see a significant global effect of the bio-inoculant on yield, meaning 0.8 kilogram per hectare. If you are not accustomed with Q per hectare, Q is a French unit, it means a decitons. So this is a significant positive effect of the bio-inoculant on yield. And we can see some various effects according to the cultivars. You can see cellule and cougar seems to show a greater effect than rubisco, for example. Concerning the grain protein concentration, we don't see any effect on this variable of the bio-inoculant. Second type of result, it's exactly the same result, the same number, I mean, but this time the results are sorted by N rate. So the black points are for the optimum N rate, and the red points are for the under-fertilized rate. As you can see, it's interesting to see that we can see a greater positive effect of the bio-inoculant for the under-fertilized treatment compared to the well-fertilized treatment. It's quite logical, but it's fine it's nice to be able to show it in the field. Concerning the grain protein concentration, we can see a very small but significant effect on grain protein concentration of bio-inoculant only for the under-fertilized treatment. And the last slide of result, this time the results are sorted by the type of microbial inoculant. The black point are for the treatment 1, which is a combination of mycorrhizae and bacteria. And the red one and the green one are also a combination either with mycorrhizae and bacteria. As you can see, it's interesting to see that concerning yield, we don't see exactly the same result for all combination of microbial inoculant. One combination, T3 treatment, seems to show a greater effect on yield. And concerning the grain protein content, we don't see any effect of one microbial inoculant or one other. So these results are still under investigation. We need to do some deeper stats because the statistics I show in the previous slide are very basic. We also have to include properly the result acquired by our colleague from Agroscope, and we are preparing a scientific paper on that. So it's still under progress. Nevertheless, I think we could draw some preliminary conclusion because this first result seems to show that point 1, we could see an effect of the tested microbial inoculant on yield in interaction with cultivar and also in interaction with nitrogen rate. And unless this conclusion won't be confirmed by deeper stat, we could expect some difference of effect between microbial inoculants type. So, and if these results are confirmed, could be quite new, given that it's really difficult to assess a significant yield effect of such input, I mean, a microbial inoculant in field condition. You can see a lot of paper in the literature showing some very interesting effect in control condition, but when you try to confirm it in the field, it's always difficult. This result seems to show that it could be possible to see something. And as a conclusion, I just want to share some thought on microbial inoculant field testing and transfer to farmers. If any, the effect of microbial inoculant on yield are small and need a strong statistical design to be assessed. So it's clear that we need several trials with replicates, several experimental sites, several years of trials, etc., etc. On-farm testing, I mean by farmers, is possible and in some extent recommended to extend the microbial trial result. Nevertheless, a rigorous method must be applied also on this type of trials to do it properly. If you use strict trials, you must have a very robust design. You must be able to do stats. You must have a lot of trials too. So as an example, it's nearly impossible to interpret the result of a single strict trial in the far end of a single farmer field, if you see what I mean. The third point is that bio-inoculants are living beings, so result analyzing must consider agro, I mean soil, climatic, I mean weather, management, I mean agricultural practices, condition. And it's also very useful after to make some recommendation to farmers to better use the product. Formulation is essential, and I think we will discuss formulation later during this meeting, and testing must ideally be done with a formulation that farmers will have access to, because the result could be very different from one formulation to another, even if you keep the same microorganism. And at the end, even if collaboration with companies are welcome to better handle the technologies, and I think it's essential. I must also underline that the testing must be done in independent field trial platforms to be able to show some independent result to farmers to help them to choose one technology or another. So I think that's the end. I hope I keep the time. I don't know, in fact.
Mm-hmm. You're very close. I appreciate it.
Okay.
You covered quite a lot of information. Thank you very much, Jean-Pierre. So just as a reminder, if you have questions, type them in the chat. We will have time for a pause for questions. But now I'm going to pass it over to Geoffrey Darbon from Agroscope and Leonidas Rempelos from Newcastle, who are going to talk about potato trials in the SolACE project.
Yeah, maybe if I unmute myself, it will be better. So hi, everyone. I'm going to Here we go. So I'm Geoffrey Darbon, a PhD student from UCL Louvain, but based in Switzerland in Agroscope. So I was responsible for conducting the field trials on potato with this inoculation of microbials. So this was the field in 2020. We had quite similarly to what was done in Arvalis, several factors. So the inoculation of microbes, also irrigation. We generated a water stress in some of the lines to see how our two varieties would answer to that. We had Pentlandel and Désiré as varieties, and also to see if the microbes would be able to compensate for any yield losses caused by water stress. So the microbes we inoculated, yeah, as Jean-Pierre explained, very different ecologically speaking. We had two fungal strains, so a mycorrhiza, the Rhizophagus irregularis, a non-mycorrhizal fungus, the Trichoderma aspergilloides, and 2 bacterial strains, Parabocolderia phytophumans and Pseudomonas brassicacearum. In the early phases of the SolACE project, greenhouse trials were performed to select some consortia, some mix of these microbial strains that were the most promising to improve potato growth. So for the first year, these 3 consortia were selected. I think we had 2 in common, Jean-Pierre, so the AMF and the Pseudomonas, and the AMF and the Parabocolderia. But we had also 1 consortium with 3 microbial strains, so the Rhizophagus, Parabocolderia, and Trichoderma. For the second and the third year of trial, we took the liberty to enlarge the trial by testing inoculation of single strains to see if we could decipher the effect or attribute the effect we would potentially observe to one or the other of these strains. We also used milligram from DCM, and this is what it looks like. So it's a microgranulate. We measured various parameters during plant growth and also post-harvest. I'm not going to present everything today. I will mainly focus on what we call in Agroscope the external defaults, which are the potato tuber blemishes. So you can see that the tubers here, they can be affected by various pathogens that develop at the surface of the tubers post-harvest, that we can observe post-harvest. So they can be caused by fungal pathogens or bacterial pathogens. Why were we looking at these symptoms? Well, because it is quite known that all these strains that we inoculate, they indeed promote plant growth, but also they can provide biocontrol against various pathogens. Each of these strains, in a direct or indirect way, can help plant health by, I don't know, producing antimicrobial compounds, also fungal cell wall degrading enzymes, or just by enlarging the surface of the roots for mycorrhiza, like Per said, and provide with better water absorption, better nutrient absorption, and so indirect plant health promotion. So we focused on these 3 diseases. So the black scurf on the left, caused by Rhizoctonia solani. I will not be presenting the results for this disease, for the simple reason that we did not observe enough black scurf. So this is a good thing for farmers, but we wanted to see if our inoculated microbes lowered the symptoms, but yeah, we cannot show any results because we had no pathogen. But we registered also the symptoms of common scab on the right, caused by the bacteria Streptomyces, the various Streptomyces bacteria. And also we registered this silver flecks on the surface. These can be caused either by the silver scurf or black dot. So these are 2 different diseases, 2 different pathogens, but they are not really distinguishable with the naked eye. So we are still producing data on this with the binocular. So here are the spore-producing structures from the silver scurf. And you see that it can't easily be differentiated from this black dot. as the name suggests, produces a black dot on the surface. But yeah, it seems obvious and not binocular, but you cannot see it with the naked eye. So what I will present today is only like a mix of these results. So silver scurf associated symptoms, both of these pathogens together. So for the consortia, after inoculation of the consortia, we see that the silver scurf associated symptoms are lowered by the consortium in green with the Rhizophagus and the Pseudomonas in comparison with the control in red. Incidence is just the percentage of the tubers that were affected by the pathogen. Another thing you can see is that the levels of infection with silver scurf was a lot lower in the variety Pentlandel on the right panel in comparison with Desiree, and that we did not observe any significant difference resulting from inoculation in the variety Pentlandel, so suggest an interaction between cultivar and the inoculated microbials. Something really interesting is that we also observe an effect, but in common scab associated symptoms. This time it's I only show the variety Desiree because we found nothing also in Pentlandel. And again, the same consortium was able to lower significantly the symptoms, but this time of common scab. Then, of course, we wanted to take a look at the single strains, and there we were kind of disappointed because in Desiree, the single strain inoculation could not lower the symptoms either of silver scurf or of common scab. The only thing we saw is that during the second year, the Trichoderma aspergilloides was able to lower the symptoms of silver scurf, but it was not the case, as you can see during the third year on the panel on the right. So yes, this is the only thing we obtained with single strains in terms of biocontrol, because the yield results, we saw that during the third year, Rhizophagus on its own and also Pseudomonas on its own, they were able to improve the yield in comparison with control. But it has to be said, no effect was observed on yield over the 3 years. No consistent effect. Yeah, it is true that in controlled condition, it is very easy. There are a lot of publications that show significant effect on yield. But once you get in the field for various reasons that we will be able to discuss afterwards, I think, it's harder to observe effects, which is not really the case with biocontrol, in my opinion. That is also why we took a look at it. So conclusions, the consortia, as the literature suggests, seem more efficient at lowering the symptoms of post-harvest diseases, especially the combination of the Rhizophagus and the Pseudomonas in the variety Desiree. Regarding the water stress, we observed no real difference. Of course, we observed the yield difference between irrigated and rain-fed lines, but no inoculation seemed able to lower the yield losses caused by water stress or any other parameters that we measured. And yeah, the yield was significantly improved only during the third year with the single strain application of the Rhizophagus or the Pseudomonas. I think I am done here, so I will end with this picture of a Desiree tuber infected by both of the diseases, both of the pathogens. And then I give the floor to Leo.
Leo, it just looks like you need to unmute and then everything else looks good.
Okay, all done. Hello, can you hear me, Emilia? You can hear me. Okay, perfect. Okay, hello from Newcastle. I'm Leo. I'm working as a researcher for the SolACE project, and within the next few minutes, I will give you a very brief outline of the results from our on-farm trials. Starting, potato trials within the SolACE at the UK network were designed to test the efficacy of AMF inoculants on different commercially available potato varieties grown in a range of commercial farms, as well as to demonstrate their potential to increase tuber yield, water and phosphorus use efficiency. Two farms, as you can see in my little map, two farms were in the east of England, and they tried the commercial AMF inoculant with and without irrigation over 2 seasons, while one farm northeast of England tried the experimental inoculant provided by the project over 2 seasons, and there was no irrigation applied. All the above trials followed a very simple on-farm demonstration strip design, while varieties have been farmers' choices, so they fit with their current cropping plans. In addition to those, we also ran what we call a controlled factorial experiment at Newcastle University in order to test the efficacy of the same commercial and experimental inoculants while comparing different commercial genotypes under limiting phosphorus conditions. Overall, when we combine the results for all 4 sites, when mycorrhizal inoculation was used, we get slightly higher tuber yields, as well as nitrogen and phosphorus acquisition from the tubers. However, we wanted to see if those differences are actually consistent within all the farms, and if there is any effect of irrigation and genotypic variation on the efficacy of those products. So for the organic farm, we have no positive effects of mycorrhizal inoculants. And there is also no variation between AMF, between the inoculants and the heritage potato genotypes that the farmer was used. On the other hand, for the two conventional farms, we identified that only when irrigation is used, we get a positive effect of the mycorrhizal inoculation on tuber yields, nitrogen, and phosphorus acquisition. And this is something quite interesting. So we wanted to see if this is actually consistent for both farms, and if there is also a considerable variation between mycorrhizal inoculation and the genotypes that the farmers used. So for the first conventional farm, inoculants increased potato yield only when irrigation was used, so it's consistent, while there is no significant variation between inoculation and the potato varieties that the farmer used. On the other hand, for the other conventional farm, there is no effect of AMF inoculation, and that's quite interesting. So this indicates that the difference between the results from the two conventional farms might have been caused by various reasons, like the difference in soil organic matter or local microclimatic conditions. And this also suggests that the AMF inoculants may not be beneficial in all agricultural scenarios, and the case-by-case assessment is required, as well as when inoculation takes place, changing management practices might also be beneficial. To summarize, our data contribute to the growing agreement that mycorrhizal inoculants have a potential to improve crop production and to slightly increase yields, mainly in relation to nutrient and water management. However, there is a variability in the efficiency of those products, and we have a limited understanding of the mechanisms responsible for this variability in on-field scenarios. Because usually these products work in the greenhouse or in the lab, but sometimes they don't work in real conditions. So these unknown mechanisms might be caused by different soil health indicators or different climatic conditions. Or it's also possible that the AMF within a commercial inoculum could fail to establish if they are not, for example, applied properly, or if they are not compatible with the crop plants that we use, or if they compete with the soils, like the soils native inoculants, like soils native communities, native AMF communities, or simply if they cannot survive the standard agronomic practices that each farmer is using. So all the above are showing actually the need that we need for more detailed investigation. So in the future, there is a need to do more research in order to answer the above questions. That's all from me. Thank you very much.
Thank you, Leo and Geoffrey. And to kind of round out our case studies from the SolACE project, we have Günther Neumann, who is from Hoffenheim and is kind of going to be giving a bit of a critique of the approach we employed. So Günther, we can see your slide, but you might be muted. We can't hear you yet.
Now it should work, sorry.
No, there we go.
Unmute the microphone. Okay, thanks for the introduction. My name is Günther Neumann from the Institute of Crop Science at the University in Hohenheim in Stuttgart. And during the project, we were mainly responsible coordinating the preliminary work, the selection of strains provided by the partners, and the testing with respect to their capacities for protective effects against drought stress and improved nutrient acquisition, which were the major goals of the project. And as we already heard, the final outcome was quite variable. I have to go further, I think. Unfortunately, now yes, it took some time. Despite of the fact what we heard that under controlled conditions, frequently we can reproducible show the beneficial effects of microbial inoculants. Here you see different fungal and bacterial strains in a large-scale tomato production experiment where the inoculants were applied during the nursery culture. When the SolACE project started, we were just in the final phase of other EU project, which was explicitly focused on these biostimulant effects and the conditions when they could work or not. In this project, we conducted more than 150 greenhouse and field trials with 3 crops and 38 biostimulant products, other products on the market, and also new developments in 11 countries. And very similar to the outcome of SolACE, the final positive results were quite variable, and we saw only in 30% of the cases significant effects. And this was the reason why we finally decided to take all these data sets produced during the project together for a final meta-analysis, yes, to get more information under which conditions we may expect positive effects. And I would like to share some of these results to you, and maybe we can learn something also for our future experiment. Yes, so this was the outcome. We had, in contrast to SolACE, we had 3 crops. We had wheat, maize, and tomato as a vegetable crop. And in general, here we see the growth or yield response over all the tested experiments. And we see a significant positive effects when we go over all experiments, about 10% growth or yield increase. But as we see, we have a strong variability between the different crops, and tomato was by far the most responsive one, which may point to genotypic differences or to differences in plant species. But a closer look also showed that particularly the growing conditions for the specific crops are probably even more important, because tomato is a vegetable crop grown at a completely under protected greenhouse conditions, or at least the nursery is conducted under protected greenhouse conditions, in contrast with wheat and maize, where we have field crops. Doing experiments under controlled conditions with the field or the greenhouse crops frequently revealed positive effects in both cases, which points in the direction that particularly the growing conditions may have a particularly big influence in this context. And this was confirmed further by looking on the different growing conditions employed during the projects, where we see the 10% yield or growth increase over all crops, but the lowest effect under field conditions, and then under greenhouse conditions, or at least in tomato plants grown in open field, but with a nursery phase under controlled conditions. Here we see the highest responsiveness, which may be related to this protected environment, which supports the establishment of the association with the microbials in the early growth phases, where they are usually applied. And during this phase, any stress conditions which can affect on one side the microbials, or on the other side the host plants, which have to provide the nutrition of the microbials via their root exudates, all stress factors which are negative on the root activity in this context are also negative for the association, and this may explain this result. When we have an established community, then we can expect protective or growth promoting effects. And also the results from SolACE pointed in this direction. These are data from the potato trials from the partners in Switzerland, where they tried to trace the different strains which have been applied to potato plants in the root and on the rhizosphere during different time points of the culture period in open field. And we see that we frequently have a rapid decline in the recovery of these microbial inoculants that after 2 months, they were in many cases almost not detectable anymore. Similar effect we saw also in other projects in this direction. This was a project with an industrial research cooperation on a microbial consortium product with multi-strains. 22 different microbial strains were applied here in field experiments or greenhouse experiments with horticulture and vegetable crops, 16 field trials, which were partially conducted really under field conditions, partially or completely under greenhouse conditions, but a common condition was the protected greenhouse culture during the nursery phase where the products were first applied. And here we see a general positive yield trend. Then in an agricultural crop like maize, with 11 field trials in 4 countries and 109 trial variants, we see this high variability we were also facing in the SolACE project. Coming back to this meta-study, looking on the different experimental conditions addressed within these experiments. Here we saw in experiments which were focusing on improved acquisition of different phosphate fertilizers, we saw a surprisingly homogeneous response in different categories of biostimulants, whether single strain inoculants or mixtures or non-microbial biostimulants, a very similar response in the range of 10%. This, of course, is a good news because this means we have a broad range of products available which can be effective. It also raises the question why we did not consider any non-microbial biostimulants in SolACE, because particularly for stress protection, the use of non-microbials is much more widespread in practice. I addressed this point at the beginning in the preparation of the project, but finally it was the general agreement to concentrate only on microbials. When we change the experimental question to really stress conditions, so experiments which have been designed to assess tolerance against drought, salinity, or cold, the picture, changes completely. Here we clearly saw that obviously the efficiency, particularly of the microbials, declined. But we also see that we have a potential benefit of combination of consortium products with complementary or synergistic properties, which may be more efficient under flexible environmental conditions. And this was also the reason why we mainly focused on consortia in SolACE. And this was reflected also in the drought stress experiments under SolACE, in the preliminary screenings, where we had at the beginning 16 different single strains or strain combinations for the screening. And here we saw beneficial effects, for example, in potato on pea acquisition in 3 out of 6 single strains, but in 8 out of 10 consortia. But here again, we see that this negative effect of stress conditions, probably particularly during the establishment phase, will negatively affect the final output. Another interesting result was that the microbials obviously have preferences to interact with specific fertilizers. And we saw in the biofactor program, particularly high responsiveness in combination with fertilizers based on manures, guano, or meat, hair, or feather meals. All these fertilizers have a common property that they have a comparatively high nitrogen and phosphate availability at the beginning. And this may, of course, have benefits because this may act as a nitrogen or P starter fertilization, which is well known to promote the establishment of beneficial microorganisms like mycorrhizae or rhizobia, particularly under limited soil fertility. And these organic fertilizers also provide an additional carbon source, since many of these microbial inoculants are known that they have a very high demand for easy available carbon sources. And finally, the improved establishment of the association under these conditions may lead to an improved mineralization of the organically bound nutrients in these fertilizers. So a lot of benefits. And the mini-gran formulation, which was already mentioned in the previous talks, which was the final formulation tested in SolACE, was a fertilizer formulation going into this category, despite of the fact that the fertilizer components were not of animal origin, they were mainly of plant origin. But they all had also a high nitrogen and nutrient availability. So now coming to the results of SolACE and an example of the overview on the potato results. Here we see the effects on tuber yield in greenhouse experiments compared with field results under drought conditions. And here we see the percent change of yield always in comparison with a drought affected control without any inoculation. So the first experiments, we were lacking a proper formulation because we started with pure strains, and therefore they were applied in artificial growth media. For the mycorrhizae, we had an alginate formulation, and this was the way how they were applied both in the greenhouse experiments and the first field experiments. And we see that the blank formulations, or also the combinations with the microorganisms, did not reveal any significant results, even some negative trends, and only the well-watered controls showed the expected benefits in tuber yield compared with the drought stress potato. And the first field results in the first years exactly revealed the same result. Only the irrigation variants were significant, but nothing, no significant effects with the consortia. In the next step of optimizing, we tried to provide better conditions for the establishment phase. And here we tested some silicatic hydrogel soil conditioners with the property to increase the water holding capacity of the substrate, and they were applied together with the inoculants at the beginning. Here we see, at least for the microbials, a certain trend, but still not significant with respect to yield results, but the only significant effect still with the well-watered controls. Under field conditions, this was not tested so far. And then the next step of development was the combination with these mini-gran organic starter fertilizers. They were usually applied by a single inoculation. In the greenhouse tests, we also used a double inoculation. And here we see clear positive trends, but still not significant, and only but clearly positive again in the well-watered control, and as we heard also in the field experiments, at least with these single inoculations, no significant effects. And now finally, in the last 1.5 years, we combined also this organic fertilizers with this protective silicatic soil conditioners. And here, at least in the greenhouse experiments, we now could see for the first time clear drought protective effects by the consortia, which showed yield increases, which were even comparable with the well-watered controls. But we are at the end of the project, and so therefore this has not yet been tested really under field conditions. So this was how these beneficial effects looked like. So we had no protective effects of the treatments without any microorganisms. We see a lot of drought-affected leaves which could not recover even after 2 weeks after rewatering. And the plants which were treated with the mini-gran formulation and the consortia showed a much lower level of leaf damage related with less oxidative stress induced by the drought treatments. And of course, this enables more leaf area, which is photosynthetically active to provide carbohydrates for tuber formation. And also field experiments from other groups show that there is a close correlation with the loss of leaf area by, for example, damage or pest attacks with the yield losses. So a positive correlation with increasing foliage loss, less yield losses. And this was finally the results on the tuber yield. Here under the controlled conditions where we have the well-watered controls, here the formulations without the microorganisms, and here the combination with the microorganisms. Of course, there are certain other effects affecting the performance. In the meta-study, we saw, for example, a clear declining efficiency of the application of microbials with increasing organic matter content in the soils. We know that higher soil organic carbon is frequently associated with higher availability of nutrient and water, and also a higher microbial activity and abundance. And of course, these are all effects which are counteracting the additional benefits we can expect from biostimulants. In another meta-study conducted by the colleagues from Switzerland, looking on literature research, they could also show that the efficiency of microbial biostimulants is usually lower in moderate climates compared to more challenging climates like in tropical or dry climates, which would be, of course, in the direction what we expected for SolACE. So finally, I would say for these microbial and also non-microbial biostimulants, we have a quite interesting potential toolbox for agricultural applications. But what we learned, obviously, we still have frequently not the exact instruction manuals for application. Open questions, particularly for microbial biostimulants, comprise the successful establishment under variable environmental conditions. Also, how to efficiently exploit potential complementary or synergistic interactions between different biostimulants, but also between biostimulants and fertilizers or genotypes, and also the native soil biota. And finally, an aspect which was not addressed at all in SolACE is how to convert this into efficient and particularly into cost-efficient application strategies, which can be integrated into existing farming practices. So thank you very much. I think this was my short overview, and I hope that we finally can enter into an interesting discussion.
Thank you very much, Günther. We have a little bit of time to do some of the questions that have come up, and it looks like people have done a good job of responding to them directly in the chat. But there's one that came up for Günther, which was what species of bacillus was used, and asking if the experiment was under NPK treatment, which I think was in reference to the earlier experiments you were discussing.
Yes, this was probably the example what we showed at the beginning. So this was a commercial product, Bacillus amyloliquefaciens FZB42, which is on the market. And this was mainly used. We had also some other Bacillus strains which were not on the market still, but this was one of the major Bacillus strains used as a reference strain during the biofactor project. So the FZB42 strain, which is well characterized, and we integrated it partially also into our early screenings to have a sort of reference.
Great, thank you. Since we have covered a lot of the questions in the chat, unless there are any clarifying questions people want to ask, I think we'll keep going, but we will have more of an open discussion once we've kind of gone through a couple other presentations now from outside of the SolACE project. So if I can invite Natalia Gubis to present a commercial perspective from PlantWorks, a company based in the UK.
Can you see and hear?
Yes, perfect.
Excellent. Thank you very much, Amelia, for inviting me to talk here. So I am representing here the company called PlantWorks, the UK leading microbial biostimulant producer. And I will be talking what products we have and how we developed them and how we tested them. So in our portfolio, we have two types of microorganisms. One is mycorrhiza fungi, and the other type is rhizobacteria. And they do really similar functions to the plant. They make them grow better, they supply nutrients, but as a commercial product, they do look different. And there are some issues with this I will be talking about. And also, they applied at very different time. The first product I will be covering about mycorrhiza fungi. The company was initiated in early 2000, and it is a spin-off of Biotechnology Institute, which overlived its mother. So back in those days, Biotechnology Institute were doing lots of research using mycorrhizal fungi, and they couldn't find a good source of inoculum. So every time they were sourcing somewhere outside, they were not performing well. So they formed a company that can produce a good quality inoculum. And for example, some of species that we currently have were developed within this Biotechnology Institute work. And here I'm showing, for example, paper where those fungi were used in reclamation of land that of soil that where channel tunnel was built, and showing how much mycorrhizal fungi can help to convert this land into arable land. We have in production 6 different species, and we produce them in monocultures. Within these 20 years, we developed, we refined all our host plants, making sure that our fungi not tuned to a specific crop type, but we just can colonize various crops. We made sure that our product is full of spores, because spore is robust propagule. And we do have very good and thorough QA methods. So we're making sure that everything that leaves our production has good quality mycorrhizal fungi. Mycorrhizal fungi, because they are obligate symbionts, they need a host plant to live, and therefore it's limiting of production methods. So our product is granular, and this may cause some issues, and probably you guys who use our product experience these problems as well, because as a granular is insoluble granules is quite difficult to apply. So for example, if it's for arable farming, some farmers can have a microgranular applicator to apply the product. But if you're talking about potato, it's a little bit more tricky. Another limitation of mycorrhizal fungi in farming is just, we wouldn't recommend to apply with every single crop in rotation. So we always recommend for the farmer to review their rotation and think when is suitable to apply. For example, if you're growing wheat and you want to treat wheat, but next crop will be oilseed rape, which is non-mycorrhizal, we wouldn't advise. Therefore, another limitation is knowledge. So farmers need to understand how mycorrhizal fungi do work and what they do, or agronomists, that's our road we choose. We train agronomists so they can explain when this product can be used. Another biostimulant we have, which is more probably suitable and interesting, and also there was lots of talk about it, is plant growth promoting rhizobacteria. Back in 2013, we followed exactly the same road as with mycorrhiza. So instead of just going ourselves and isolating species, we went to well-known researchers and asked if they have any good species that they would like to commercialize. So their knowledge is not just used for academic purposes, but also is used for practical reasons. And we do collaborate with James Hutton Institute, a Spanish research group, and French research group, for example. And we have 14 species in our collection. So they were all tested by researchers, but what we had to do, we had to make sure that we understand how to produce them on a mass scale. So, and what is the shelf life of the product gonna be? So we produced various grow curves, so we know when them harvest, and also we made sure that shelf life is suitable for, as long as suitable for the farmers. So we've been doing this lab work, and after this, we took this species into like small pot trials in our greenhouse. And what we thought, if we combine more species, it will be more beneficial. Basically, the more the better. But it turned out was wrong approach because certain species will benefit certain crops. Here, letter coded are different bacterial species. And you can see, for example, species F is beneficial for crop 1, but not so beneficial for crop 3. And that's us prompt to test every single species we have of bacteria on for different crops, and then make a consortium that will fit for certain crop. For example, we do have a special consortium for wheat, special consortium for potatoes, or special consortium for oats. We also had to determine what is the optimal CFU number, because we do not, and probably we won't be able to dominate population, but we would like to modulate rhizosphere population enough so plants have benefits from bacteria. Here you can see how we do test in various different crops to the inoculum. And I couldn't resist to insert this picture, just showing that effects people have are actually from microbes, not from any additives. Here is plants growing in coir, and coir is quite harsh inoculum, a harsh environment for the plant to grow. And what we can see here, this row is this column doesn't have any microbes. This one will have mycorrhizal inoculum, but autoclaved, basically killed biology. Plant growth promoting rhizobacteria. This is number 3 row. Number 4, mycorrhizal fungi, and number 5, combination of 2. So again, what Günther was telling is just we need to make sure we know which species are really grouped to combine so we can have a synergetic effect. And here you have inoculum, but that inoculum doesn't do much. And when we go, after we complete all lab trials, greenhouse trials, we go on the field scale. We collaborate with farmers, we collaborate with various agronomy companies, research institutes. Here is an example of our monitor farm. We do use QGIS method, where we have different strips with treated and control. And we do take we select a various data point so we can write a proper statistical analysis on this. And this is after we determine which species are good for which crop. It's good to have yield increase, but we looked further and we decided what if application of bio-stimulant can offer reduced nitrogen input. For example, farmer can use less nitrogen, but have the same yield or maybe even increase. And this data is in a way with correlation with Jean-Pierre he was mentioning. When you reduce nitrogen, actually you have a better effect of biology. So you can see here where it was control, 100% nitrogen applied. And if you reduce down to 55%, you have actually yield increase compared to control. So less nitrogen, you add biology, and you have even higher profit. And and the previous was and previous slide was on basically this is in prices in 2020. So fertilizer prices 2020, farmers can save 202 pounds per hectare if they apply inoculum and reduce their nitrogen input. And as you're aware, now nitrogen is much more expensive. So farmer, this cost numbers will be even more impressive. Thank you very much. And if you have any questions, happy to answer.
Thank you very much, Natalia. Yeah, we have some time for questions specifically for Natalia before we move on, which none have come up in the chat so far, but I do think it's really interesting what you just noted, that fertilizer prices are increasing a lot. Have you noticed that farmers are asking you about trying to reduce their?
Not just farmers, agronomists as well. We already train the major agronomy companies in the UK. We do sell lots of inoculum to Hungary, for example, and we do, yes. That's another issue with British farmers. They're really conservative, but now they don't really have choice because they don't have, either fertilizer is too expensive or they can't buy it. So they're really looking for alternatives. But we conducted this trial in 2020, even before all this fertilizer story happened, started.
Yeah. And Julia has asked a question, because you mentioned that the application method can be tricky. What application methods do you recommend?
Right. For mycorrhizal fungi, because it's a granular product, we do recommend to use with cover crops. So when cover crops go in the ground, the farmer either buys pre-blended, because some suppliers can blend mycorrhizal fungi with cover crops and drill, or they blend themselves and drill and maintain in a rotation within 5-6 years until they grow, for example, brassica or some other soil disturbing crop. With bacteria is much easier. You just in the springtime put in a tank and go and spray soil, because there's a rhizobacteria living in the soil. You just spray soil, literally beginning of March soon.
Yeah, interesting. Because I think Julia's follow-up question was about, is it important that they
yes, for mycorrhizal fungi, yes, I missed this point. Really good question. With mycorrhizal fungi is important at time of drilling. So if it's almost impossible to get mycorrhizal inoculum when plant is already growing, you have to use either inoculate companion crop and just plant. With bacteria, you need to have rules already established. That's why we recommend to spray when plant is emerging or early March for winter crop. For mycorrhiza, definitely in a close contact. Otherwise, they won't be able, they can't live without host plant.
And I imagine this will be something we come to in the discussion as well, is the difference between application when you're doing a controlled experiment versus trying to apply it across an entire field as a farmer. Is a bit of a different, a different question. Well, thank you very much. Unless there are other questions specifically for Natalia, I think we'll move on to our final case study. Yeah. Oh, Geoffrey, sorry.
I just had a question. I'm not sure I understood right, but you said that with mycorrhizal inoculants in particular, you are using cover crops as kind of nurse in the field prior to the cash crop, in order to have enough inoculants ready when you plant the cash crop.
Yes, that's absolutely right. What we advise farmer to do, use cover crop as a vectoring to vector mycorrhiza into soil. So for the following cash crop, mycorrhiza is already in the soil. But we also advise all these kind of regenerative principles, have ground covered all the time, try to grow a non-mycorrhizal crop as less as possible. And I was showing an example when you grow brassica crop, that is a good point after this crop to inoculate. You inoculate with cover crops, and then you don't have to inoculate again until you grow brassica again. But you have to look after mycorrhiza. If you harvested your crop, put straight away either catch crop or cover crop, something, so to have this continuous host plant growing in soil.
Thanks.
Thank you very much again, Natalia, and everyone for the questions. And oh yeah, Miriam, who is our next speaker, has a question as well, which maybe we'll get to in the discussion. But our last case study is from Miriam Silger, who is based partially at Newcastle University, but also with INOQ in Germany, and is going to be talking about an example of using mycorrhizal fungi in experiments in INTERFUTURE. So Miriam, I know you had some technical difficulties earlier, but are you able to join us?
1 second.
Okay.
Oh man. Oh. Okay. This is so confusing.
So far, you're doing it though.
Okay, did you see my screen?
Yep, see your screen. It's not in presentation mode yet though.
Yeah. Okay. Hang on.
It's still not.
It's not in presentation mode.
Miriam. I mean, it's probably on another screen, is it?
It is.
You just have to switch in your presentation settings, just switch the screen to whichever one your Zoom is on.
Mm-hmm.
Yeah, and I can't read German, so I can't help you there, but I know it's one of these ones you're looking at.
It's possible.
Has it got main screen and it'll have like your monitor and your other screen. You just choose the one you want it to present on. Oh, it's the, I can see it says Bildschirm or something like that. Okay. I got it. You got it.
Is it now working?
Okay. Yep, you're full screen now.
Perfect.
Perfect. Okay. Right. So Amelia, thank you for the introduction. I did my project with INOQ and Newcastle University, and it was basically about how we can optimize arbuscular mycorrhizal fungi with wheat. So we've heard a lot about mycorrhiza, so I think I don't need to introduce them to you. We had a international training network, so INTERFUTURE is the name of that project. And the core of my project in INTERFUTURE was 2 field trials that we conducted in 2017 to 2019, close to Newcastle, so at Nufferton Farm, where also other trials of SolACE were conducted. And these soils were quite unique because they had a very low phosphorus index, so almost no available phosphorus there. And here on the right-hand side, you see a block. So we had overall 4 blocks in an area of 0.3 hectare, and half of this area got inoculated with inoculum that I will talk about in a second. So one block was composed of a field that was either sprayed with pesticides or not. And we had 2 varieties that were either from a conventional breeding background or from an organic breeding background. So we tried really to have contrasting conditions here. And we tested 4 different fertilizers, so no fertilizer. Then we had mineral nitrogen at an application rate of 170 kilograms per hectare. We had biogas digestate and farmyard manure applied, and ended up with 32 conditions. For biomass and mycorrhizal root colonization, we monitored that over the growing season and sampled at 5 key growth stages. Later, we also looked at diseases and measured SPAT. And at the end, we looked at yield parameters and how the grain quality was affected by the different treatments. The field application was done with INOQ Advantage, actually also the other one that I will talk about later. This was formulated as a root powder, and we wanted to apply 100 grams per hectare of this root powder, which is quite a low dosage already. We used 150 grams and mixed it with 15 kilograms of sand for the field application. And then, mix it in a cement mixer or in a bucket and filled it in a sprinter for ST provided by Horsch. We wanted to, like Julia now asked, it's quite important that the strains colonize the roots. So we wanted to facilitate that by placing the inoculum below the seed band. And therefore, we got this specific machine provided that could drill the seeds and then place the inoculum band 2 centimeters below the seeds. And that in that way, we would have the simultaneous application of seeds and inoculum. And what I forgot to mention, all the inoculum was composed of Rhizophagus irregularis. I think that the main species that was also mentioned today in the presentations. The main effects that we got from the inoculation in the field was that we had no effect on mycorrhizal root colonization. Here, the major factors were really the variety that we used, where we had higher colonization in Skyfall, so the modern variety, all the way through the growing season in both years. And also, I didn't show that here, but the fertilizer was very decisive on mycorrhizal root colonization. In terms of yield or growing output, we had a minor effect on total biomass in both years. So with the inoculum, we could increase the total biomass in 0 fertilizer conditions. And without, with mineral nitrogen, we actually decreased the amount of tillers and therefore also reduced the amount of total biomass that we harvested. However, this effect was not visible in grain yield. So when we look at this figure here, we really talk about straw biomass, and that's mostly because of tiller numbers. These were the only effects we found with inoculum in the field. There were quite some challenges that we realized during the field application, but also afterwards. For example, the dosage that we applied was quite different in the 2 years. And this was caused because the production of inoculum is done basically under environmental conditions. So in INOQ, they produce these inoculum in tunnel tents. By now, I know this will change in the future, but therefore you have very different amounts of propagules in the batches, depending on the weather conditions during the production. And if you calculate that back to the seed rate, it's quite, it's very different how many propagules you have per seed. And I think this is maybe something that we need to discuss later on how we can adjust that in the future, or if this is just something that needs to be changed during production. Another problem that we face were native mycorrhiza. So this picture was taken from a plot that was not inoculated straight after the winter. So we did have already mycorrhiza colonization in tiller and at tillering, but also at seedling stage. So very early in the growing phase. And therefore, we couldn't say if the mycorrhiza that we found during colonization assessment were actually affected by the inoculum, or were actually represented by the inoculated strains or by native mycorrhiza. And therefore, we came up with a digital droplet PCR assay, which is known to have very high sensitivity for small amounts of molecules. And we designed specific primers that would only target the strain of Rhizophagus irregularis that we applied to the field. So here you see samples. All the ones with the plus were inoculated, all the ones with the minus were not inoculated. And usually you would expect nice positive clouds like you would see here in the sand and INOQ positive control. But the only signals that we got in these digital droplet PCR assays were some random lonely droplets. So you cannot really say that the inoculum established. And this was basically the outcome at growth stage 32. So that's in May when the plants are already have grown quite a lot. So when the inoculum or the strains are not in the roots by then, I would say they will come later in the season. The question is then, why did the inoculum not established? I can come up with a couple of potential explanations. For example, it could be environmental conditions, the climate, the soil, which was quite heavy, loamy soil. And it's the question if the mycorrhizae that are produced in sand beds can adapt to these very different soil conditions. Then, as I mentioned, the competition with native mycorrhiza, but also experimental factors. For example, the crop protection treatments that we applied could have had detrimental effects on the mycorrhizae, but I already showed you the graphs from the varieties, and it looks pretty much the same with the crop protection treatments, but here we don't have big differences when we apply the pesticides or the fungicides. So this should not have hampered the inoculum to establish. One possible explanation could be that the inoculum got inactivated during transport. We did take care of that, but you never know, right? So what we did in the first year, in 2017, was that we set up a couple of pots with sand and the inoculum and some wheat seeds on the window bench and let it grow for 6 weeks. And then we stained the roots and looked if they were colonized. And back then, beginning of my PhD, I thought that we would actually find colonization. But now that I look at this picture here, we see the small root piece that is fully colonized with vesicles. And I think that's actually a fragment of the inoculum itself, whereas the root besides this tiny piece here is empty. So I think also the inoculum that we set up in the small pre-trial before we started in the field, or actually simultaneously while we started in the field, was not active. Another explanation could be the application method. So now we heard from Natalia, the timing is very important. And since we used winter wheat, we applied the inoculum in autumn. So it was dark, it was cold, and therefore maybe that was hampering the inoculum to establish, and therefore ultimately also leading to the competition factor, whereas the native, where it might have been more adapted to these conditions. Again, the dosage, again with competition, could have been a factor, but also the formulation. And here I took a picture of the hopper that we used to drill, where we filled this 15 kilos sand and inoculum blend in. It basically vanished in this dimension, and we already used the smallest sprinter version that we got from Horsch. So I think in the future applications, this dimensions need to be overcome somehow. And also something we noticed was that the inoculum, when we mixed it with the sand, was sticking to all sorts of surfaces. So for example, on the gloves, on the buckets and everything. So we needed to do something about this root powder as itself, as the formulation. And for this, I decided to create a seed coating, because I thought if the inoculum is stuck to the seeds, you can create a much closer environment of the seeds and the root powder, so that the colonization would happen much earlier. And for this, we did a cooperation with the University of Applied Science in Bielefeld, and also with two cellulose industries that provided samples of their compounds. For this, I chose hydroxymethyl celluloses as the binder substance to stick the inoculum and also the filler to the seed. So the filler in this case is the equivalent to the sand that we used in the field, because we wanted to give some volume to this inoculum. And here we decided to use ultrafine cellulose and pea fiber. And then the additive, so the active compound of the seed coating in this case, of course, was the inoculum. Although we didn't have a pen coater, we came up with quite nicely coated seeds. And since we wanted to exclude the competition factor, we used sterilized field soil from the same plots where we conducted all the field trials, and filled this in pots, and did a small greenhouse pot trial, which was conducted by a master student, Han. And here we compared coated versus non-coated and inoculated versus non-inoculated. And the first thing that we could observe was that coating delayed and also reduced germination, and which ultimately also decreased biomass. So we ended up, when we coated the seeds, with lower shoot biomass. Interestingly, the same happened with inoculation. So inoculation, either in the coat or in the soil, reduced germination and also reduced biomass. So we had different coating treatments. And we also had 6 samples where we just added 1 gram of inoculum into the soil. And with both, with the coated ones and also in the soil, the inoculum, just the presence of the inoculum reduced biomass and also germination. Interestingly, this happened without any mycorrhizal root colonization. And so again, we had an inactive inoculum, which was potentially placed in an antagonistic environment. Here I mentioned the soil sterilization, and I want to encourage you, if you do pot trials with microorganisms, take care of the pasteurization method that you used. I found now that people use a very slow pasteurization method for soils. For example, over I think 6 hours at 60, 70 degrees in the autoclave or something, and we put it straight in the oven for 8 hours at 120 degrees. That might have changed the soil biochemistry quite a lot. So for future considerations, and also maybe for the discussion that we have later on, the question is, where should we start testing biostimulants? Should we start in the field or in the greenhouse? Because we started in the field, but at the end didn't have an active inoculum or large effects. But on the other hand, of course, field research in this context for wheat particularly might be most relevant, and therefore you have to have an active biostimulant that you might have to test before. So yeah, that's a big question here. You need to pre-investigate the native mycorrhizal communities. Potentially you don't even need to apply microorganisms. And here I want to encourage future studies to trace the introduced strains, because based only on colonization, you cannot say if a biostimulant established or not, especially with mycorrhizae. And this might be quite challenging because there you need to look at strain level, and particularly with mycorrhizae, that's quite complicated. How to determine the optimal dosage? I mentioned that before. It's crucial to state on the products how many propagates you have. So also in experiments, you can state how many propagates you need to establish a working mycorrhiza community that will give you certain effects, which brings me to the last point, which is economic feasibility, which is really essential at the end for the farmers. So I think it's important to include available machinery so that people don't have to purchase new equipment. And in this way also optimize profitability so that the effects that we see in the field are really lowering the costs for the farmers at the end. Based on the results of my PhD, I think there's a lot of more research needed, and I think there has been a lot of progress, but really to especially work on this economic parameter and also in consideration of native mycorrhiza, there's still a lot of potential. Thank you for listening. And if you have further questions, we can discuss later or contact me. Thank you.